Okay, I'm going to butt right in, and I designed my talk to be rather provocative. So the title is Stress Sensitivity of Our Crop Plants, but my main theme is we are ourselves to blame. And to go back to the root of the problem, we have to go back actually quite a long time ago. And so the next slide is a slide that you probably might be aware of or might have seen. It's an indication of all the regions where we think agriculture originated. And those areas, they are thought to have developed agriculture all independently. So it was a development in our human evolution to go from hunter-gatherers, and we still can find, of course, groups of people that still live by hunting, gathering, but most of the ideas that we have is that those are people that live under conditions that are not very prosperous, do not have a nice, lush vegetation to pick from. But when you think back 10,000 years, then the situation was, of course, quite different. I mean, there was no competition so much of people that were more advanced. And so those hunter-gatherers, where would they go? They would go to regions that were quite favorable for growing plants and finding items to pick. And so that is actually one of the characteristics of those areas where agriculture originated. You can see in the next slide that they were areas with quite abundant water supply, so either precipitation or a river area that provided water throughout the year. And in the next slide, you can see that also the soil fertility in those areas always was quite good. And so the vegetation would have been quite nice. If people then started to pick their favorite food as a hunter-gatherer, they picked plants that were adapted to sufficient water, sufficient nutrients, and they were actually quite fertile. So those plants that were the originators of our current day crops, they were selected from areas that were actually quite unstressed. Plenty of water, plenty of nutrients. And these areas are actually still the main areas of our agriculture. So this is an indication. The density of the brown, the darkness of the brown indicates the intensity of our farming production. So what we are left with after these three slides is that the plants that we selected for and started using in agriculture, they were from areas that were virtually stress-free. Second thing that these farmers, these original farmers, these all-day farmers started to do was convert from picking plants that they could find in the soil, on the dirt, to actually plant them for next year. So how do you do that? What is the best practice to do that? Is you sort of get rid of the competition and you plant your selected seeds in a plot which is free of other plants, free of competition. If you are successful in scraping away the competition, have an open soil, and then have a successful crop in the next year, then what you're doing is you're selecting plants which in Grimes plant strategy classification, which is a concept in plant biology that is already quite old, but it's still being used by ecologists, and that is an indication of in which category a certain plant type you can find. And if we start in the lower left-hand corner, which is indicated with ruderals or colonizers, then you are dealing with plants which are thriving under stress-free conditions, under competition-free conditions, and they are thriving in disturbed circumstances. So exactly what those first agriculturists were doing. They were scraping away the other plants, shaking up soil a little bit, disturb the vegetation, and there these plants were thriving. So the other two categories are completely different. So you have on the top, you have the competitors. So these are the climax vegetation, the latest stage in succession. So multi-year plants, and the trees are belonging to that. And that also has a very strong implication on how stress resistant our current crop plants are. The right-hand lower side, that's the area where you find the specialists, the ones that are dealing with stress. They avoid competition, but they are specialists in such a way that they can deal with droughts, they can deal with salinity, salt in the soil, they can deal with flooding, they're sometimes aquatic even. And so those specialists are a category by themselves. But we are selecting our crop plants from a particular type of plant with a particular strategy. And those are adapted to low stress, no competition, and disturbed environments. So the fact that we selected them from those strategies has implications for the stress resistance of our crops. And to explain that, it's a little bit a complicated diagram. But what is indicated here is from the left to right, the vegetation succession. Yeah, so you start with colonizers, rudderals, the R strategist in Grimes' triangle, and you move along in the succession until you get to the climax vegetation, and those are the competitors, the ones that are dominating all the other plants and hold their place. On the bottom, you see soil microbes, but they also follow that succession. And the interaction that the soil microbes have with the vegetation is indicated by the arrows. What you see on the left side, the colonizers, the pioneers, and their soil microbes, they have red arrows influencing each other. And that's a negative influence. So the colonizer plants are very bad. They are fighting their microbes. They actually try to get rid of them. They toxify the soil for the microbes that are living there. And the microbes that are surviving are actually negative for the next generation. So if you do an experiment, plants that are colonizer, you grow them on a certain volume of soil, and then try to plant the same plants the next year, they're doing worse because of the change in the composition of soil microbes, bacteria and fungi, that are actually negatively influencing the growth of the plant. So that's a negative feedback loop. Yeah, it wears each other down. If you go to the mid-successional stage, it's more or less a neutral relation that the soil microbes have with their vegetation there. And it would have the capability of staying in that stage were it not that the competitors are taking it one step further. They are mutually beneficial. So the soil microbes that a competitor is attracting to itself is actually stimulating the growth in the next season. And also the bacteria and fungi that they select, they are benefiting from the plant that's growing on that volume of soil. Furthermore, there is a negative feedback to the previous successional stage. So the soil bacteria under a forest with only competitors, only with multi-year trees, they are actually suppressing the mid-successional stage. And the soil bacteria in the mid-successional stage is suppressing the pioneer stage. So ecologists have even claimed that succession is not so much driven by the plant species composition and the invasion of competitors, but by the soil microbes that make the soil suitable for the competitors and unsuitable for the colonizers. What does it mean? So if you realize that our crop plants are selected from those colonizers and pioneers, they do not care for their soil microbiome, and they do not care about their trait in helping the plant to grow and succeed in their fitness, in producing seeds or crops or whatever it is. And that is point 2 that I want to make. Now the last point. When breeding, what do we focus on? In our breeding programs for better crops, we focus on increasing yield. Yeah? And increasing the yield is enabled by providing the plant with enough fresh water, more than enough nutrients. And so what does the plant respond to? It doesn't need to invest in a root system, and it also does not depend on any of the soil microbiome, the fungi and the bacteria in the soil, that might help him to gain a better efficiency in taking up nutrients. Yeah, because the farmer is providing it. So since that was not a breeding target, what you see in this diagram is the root system has diminished. Yeah, it's becoming scarcer. Not much biomass is invested in the below part root system. It has become less branched, so the efficiency of uptake has actually decreased because it was not needed, because the farmer was supplying all the nutrients in the water that the plants were needing. What also was, and that's only a recent realization, what also was an unintended breeding outcome was that actually the amount of investment of a plant, which you have to realize, 15 to 25% of all the photosynthesis that a plant is performing is being put in the soil, yeah, as compounds that leak out of the root system and is feeding the fungi and the bacteria. And that is also decreased under our breeding goals. Yeah, so we didn't pay attention to it. It was lost at the benefit of a higher yield. But now we are dealing with abiotic stress. And abiotic stress, it can be drought, it can be flooding, heat, or salinity. And this is a projection for the future years of where the crop yield is going. So the productivity in all these countries is mostly going down. So we see that global change changes the amount of abiotic stress that plants are going to experience to the point where they stop growing maximally and our harvest potential is going down. So we are now dealing with plants by the very nature of it, and by the way, we have breeding goals in our generation of what we call elite cultivars that actually make them much more sensitive to all these potential threats in abiotic stresses. So one of the things that we focus on is the dealing with a particular stress called salinity. We are here in a delta, and globally the salinity stress in deltas is predicted to become a major factor in crop productivity. And one of the things you have to realize is that most of the river deltas are the areas where most of the population and also most of the traditional agricultural focus was. So all these dots on this map are deltas, and quite a number of them are under threat of salinity. So we are in the Netherlands, which is a delta, and we also have maps of increased salinization threat. And you see the north and the southwest, which I've circled, they are the ones that have an increased salinity risk. And bad luck, because those are exactly those areas. Also, the Noordoostpolder is one of those areas where we have focused our seed potato culturing. The soil is okay. The conditions are just optimal for seed potato productivity. And as you can see, the more blue, darker blue it becomes, the more profitable agriculture is in those areas. So seed potato cultivation is one of the most profitable ways in agriculture to earn your money. And so those areas, as luck might have it, are the ones that are most prone to salinization and run a risk of being soiled. So I made my point. So we have very stress intolerant plants, and we are dealing with a need for producing strategies in which we can still grow these highly yielding crops. What we can do is, with the realization of the history of all these crop plants, we can start to sort of redefine our breeding effort and our breeding goals. And one of them is to actually pay attention of what a potato is really poor at. So if you look at a potato, it doesn't invest so much in its root system. It's not very dense, it is not highly branched, it's actually quite inefficient. It's also very shallow rooting, and so for drought stress, it is not very suitable. So one of the breeding goals, it's a big project that we are working on now, is trying to actually not focus so much on salt sensitivity and making them more salt resistant by per se, but focusing on the background of why we lost this stress tolerance in the first place. And one of them is to put focus back on the root architecture, which could be improved. One of the realizations that you also have to have is that traditional breeding would have as some sort of immediate gut response. Okay, let's see what kind of traits we have to introduce in the plant. Yeah, what are the markers? Which genes are really essential for this salt stress tolerance? Now, salt stress tolerance is a very multigenic property, but if you're not a specialist, the only way to deal with salt stress is to keep it out of the leaves. Yeah, only the specialists, and they were already mentioned, a couple of them, only the specialists can deal with a high salt load in the leaves themselves. For the rest, the traits for salinity tolerance have to be sort of sought after in the properties of the root themselves. Expose them less on a per area basis to high salt concentrations, and select for those varieties that have a tendency to keep salt out in the first place already. So that's one. Yeah, it's a somewhat circumround, indirect approach that I'm proposing here. And the other one is to go for one of the other properties that were neglected or actually negatively influenced by breeding goals, and that is the relation with microbial organisms. So bacteria and fungi. On the left, you see an example where we show the effect of exposing plant roots to a Bacillus mycoides strain that we have isolated in soils. And what you can see is that it stimulates the production of root hairs. Yeah, so thereby increasing the efficiency of uptake of the plant tremendously. And this is a non-traditional symbiont. So we all know the symbiosis with mycorrhizal fungi, the association in which the root is actually investing a little bit to stimulate the growth of fungi, that doing the uptake. We also know the rhizobial symbiosis with legumes, binding nitrogen from the atmosphere and making it available to the plant in a form that it can use. But these are symbioses that are now coming up the last 20, 25 years, that we start to realize that these unsuspecting microbes and the way in which they do it, we do not fully understand, but it's just making use of manipulating the plant in developing in ways it can already by itself do, but they don't do it because we have bred out the right response to certain conditions. So exposing them to, on the left side, bacteria, on the right side, very suspicious Fusarium species, but it's actually stimulating under stress condition the growth of the root biomass, so also countering the effect of salinity in a way. Fusarium, of course, is not a good name to market your product with because it's also a known pathogen genus, but this one has no pathogenic properties. It only stimulates under stress conditions the right organs to expand more. So, and the last one is this. This is a big project funded by the European Community, in which we have scanned 250 genotypes of potato to see if there were still genotypes that actually cared for their microbiome around the roots. So we scanned them first for compounds, the amount of compounds that they exude through their root system, and are now in the process of analyzing the composition of those root exudates and if they have indeed a more positive relation with their microbiome. It's a long shot. I'm doing it together with my microbial colleague, Joanna Falcao Sales, and we think that even though in the breeding program we lost most of the care for the root microbiome in the potato itself, we hope that there are still traits to be picked up from that large collection. So what I try to do is give you a sense of the mechanisms by which we lost in our crop plants a large part of the natural resistance to stress conditions that were present in the progenitors of our crops, and that we might have to rethink a little bit our molecular gene-for-gene approach to bringing back that resistance, but also go in a different way and try to make use of the ecological interactions that are also available as goals. Thank you for your attention.